The numbers yet, are they? Actually, I'm not telling you the numbers yet. I mean, I know what they are, but tough. So, you gathered it's probably going to be a linear relationship over the first 30 minutes, and that's correct. You're going to be graphing something that's going to have a positive slope for the first 30 minutes. So you'll just pick 2 points on that part. In fact, you could even draw a line to make sure that you know what you're talking about. Pick 2 points on that line, and then calculate the slope. And in the slope, what always goes on the top?
Y.
Yeah, y on the top. On the bottom? X. What are the units? Micromolar over minute. There you go. Do not forget the units in your slope, or thou shalt be sad and uncredited. Got it? Every slope has units. I hope in every lab report that you turned in yesterday and today, that when you calculated the slope, all of those slopes were in milliliters per minute, because that was the y unit over the x unit. Okay? And if they weren't, you're losing points. Do it once? Okay. Do it twice? What the? All right, so the question is, we know after 30 minutes something is going to change. What could change about the graph after 30 minutes? It could start to go down. Okay, that's possible. So option 1, maybe it does something like that. What would explain it going down, since that's part of the question? Explain why it changed after 30 minutes. What is this maltose? Substrate, product, what? This is a product, right? So why would the product go down? Again, this is a buildup of product, right? It's like we're making cars. And we suddenly got to a point and cars start disappearing. Why would cars start disappearing? Hmm? Well, if you run out of substrate, won't you still have all the cars? Like, if I've got stuff to build 100 cars and I get to 100 cars and I have no more stuff, won't I still have 100 cars? Why am I having less cars? Yeah, they're being used somehow, right? So if this thing starts going down, that tells you that the maltose is somehow being used. Now, would that be a surprising result, since we're talking a test tube that has nothing in it but water, the substrate, and the enzyme? This is not like we're talking about whole cells in here, correct? Is there any reason why maltose would be used where you just have this enzyme? Would that make any sense? Not so much. Odds of this curve actually starting to curve down are pretty low. That would be very unusual. What else could happen? It could accelerate. Maybe it goes up like that, right? What could cause it to accelerate, to get steeper, for the rate to become faster? Hey, what speeds up the rate of a reaction? Catalyst. So maybe, what did we add at this point? Maybe we added more catalyst, more enzyme. Maybe for some reason the curve spikes upward because you added more enzyme. That would make sense, right? Okay. Any other possibilities for what this curve could do after 30 minutes? It could flatline. There you go. So why would it flatline? You already told me the answer, by the way. There's no substrate left. Yeah, if it flatlines, that means you're out of substrate. So did the enzyme change at all? No. It's just like your car factory is out of car parts. You can't build any more cars. Is the factory destroyed? No, the factory's still waiting to go. If you got more car parts, the factory would kick back in and make more cars. So if it flatlines, that means you ran out of substrate. Okay? So that's pretty much the 3 options, right? If it's not going to stay the same, flatline, go down, spike upwards. Going down means your product is being used for some reason, which would be weird. Spiking upwards means for some reason the reaction is going faster, so maybe more enzyme was added. Flatlining means your reaction basically stopped. You probably ran out of substrate. Make sense? Any questions so far? All right. So, that was part A and B. Part C, draw and label another line on the graph to predict the results if the concentration of enzyme was doubled. All right, so what will we see if we doubled the concentration of enzyme? in this first part here, what would happen to the rate? It would increase, which means the slope of this line needs to get, yeah, higher, steeper. So if we double the enzyme, maybe it goes like this, right? The rate is faster. You doubled the size of your factory. You can build cars twice as fast, correct? Now, if it leveled off and ran on a substrate like this, would this curve do the same thing? Yeah. Would it do it higher, lower, same level, what? Higher. Remember, this is total amount of product, right? So where would it level off? Yeah, it would level off at the exact same level if you were running out of substrate. All right, it would just get there faster. It would probably take, half the time. And boy, the way I drew that, about 15 minutes. If you doubled the amount of enzyme, you'd probably run out of substrate twice as quick. And then, of course, explain your predicted results. Why did the curve get steeper? Well, what did that change about the reaction? Increase the rate of the reaction. Therefore, the slope should be steeper. Why did it level off at the same place? Did you change the amount of substrate? No, right? You had the same number of parts to work with. You just ran out of them faster. Question? It will go up steeper. It will take less time to level off because you're out of substrate, but the total amount of product you make will be the same. If you start with the same amount of substrate, the maximum possible product you make has to be the same. You can't make more, you won't make less. You'll use it all. All right? Yeah, just faster.
Was that the first
Hmm?
Is that all we need for the first thing?
For part C? Yeah, you drew. Oh yeah, you're going to label that line saying, so you would draw an arrow saying with double enzyme or something like that. You drew, you labeled, you explained. Sounds like part C. Part D. Identify 2 environmental factors that can change an enzyme-mediated reaction. Time? That's not an environmental factor, really.
Temperature.
Temperature! Ding, 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 ding. Didn't you do a draw, label, and explain to answer exactly that? What was the other environmental factor in your draw, label, and explain besides temperature? pH was the other one. You remember I even told you to draw the curves, didn't I?
Yeah.
And then explain why they happen? Go back and look at that DLE. That is part D. It's exactly where I got it from. I got that question for that DLE number 6 that you did a couple weeks ago from part D of this essay. You've already answered it. But just to review, when the temperature is too low, what happens to a chemical reaction? It slows down. Why? Slower molecules, meaning less of what kind of energy?
Kinetic.
Less kinetic energy means the molecules move slower, they enter the enzyme slower, the reaction happens slower. What happens when the reaction gets too hot?
Oh, it denatures.
Denatures the enzyme, so the reaction stops in a hurry. Yep. What about pH? What happens when the pH is too low? Looking for a word starts with a D. It denatures the enzyme. What happens when the pH gets too high? It denatures the enzyme. There you go. That's all you got to say. Discuss how each of these factors would affect the reaction. That's what we just did. All right. Low temperature slows down a chemical reaction because there's less kinetic energy. High temperature stops a chemical reaction because the enzyme denatures. Low pH and high pH stop an enzyme catalyzed reaction because the enzyme denatures. All right?
Do we get more points if we mention the structure, the protein structure?
It's possible. Hey, I haven't looked at the answer key recently, so possible. Are there any other environmental factors that you could think of?
Pressure.
Pressure? Not so much. Salt concentration is the third one, actually, if you wanted to discuss that. But you know what? Stick to temperature and pH. You've answered those already, haven't you? Yeah. That's one down. Shall we go on to the next one?
Yeah.
Yeah. ATP and GTP. Describe the structure. Hey, wasn't that in the draw, label, and explain you did? Describe the structure of ATP? Huh, where do you think I got that question from? So what is the structure of an ATP? Three phosphates, ribose sugar,
And
and adenine. Yeah, adenine, a ribose sugar, and 3 phosphates. Hey, what's the structure of GTP, just to be complete? How many phosphates? 3 phosphates attached to the exact same sugar called ribose. The only difference between ATP and GTP is guanine instead of adenine. Yay! Explain how chemiosmosis creates ATP. Dun, dun, dun. We just went over that. We went over that at length at the beginning of class, didn't we? What's the membrane in the mitochondria? The inner membrane. What's the inner membrane of the mitochondria have in it? An electron transport chain. That's correct. What delivers electrons to the electron transport chain? Electron carrier is called NADH and FADH2. Yeah. As the electrons travel down the electron transport chain, what do the cytochromes of that chain do? They pump protons from where to where?
From the
Matrix to the intermembrane space. That unequal distribution of protons is now called a electrochemical gradient. Yes. Now, we did all that so we can really answer the question, how do we make ATP? What are the protons going to do? They're going to diffuse where? They're going to diffuse through what enzyme? ATP. Through ATP synthase. They're going from what concentration to what? High to low concentration. That movement, that diffusion is called chemiosmosis. All right? The flow of protons through ATP synthase is called chemiosmosis. It was an arrow. Oh, hey, 6th period. Did I pass this out to 6th period?
No.
Oh, yeah, I forgot to. It's one of your assignments you're supposed to do.
Oh, that's nice.
I know. Chemiosmosis. It's this arrow right here of the protons flowing through ATP synthase. It's right there. You're supposed to label it, color it a pretty color. So 6th grade people.